So I give the context of the project I'm going to talk about today. It's a FR-AgENCODE project, like French Animal Genome Encode. So it's in the context of farm animals, and it aims to study the genotype-to-phenotype relationship to both better predict, like doing GWAS or genomic selection, and also to better understand the molecular mechanisms. So we actually, to do that in the context of farm species, which lie really behind human and mouse in terms of functional annotation. So we need a better functional annotation. And this is why there has been this FANG initiative. This is an international initiative. And on the top right, you can see the map with the different labs involved. And actually, the FR-AgENCODE project was one of the first pilot projects of this initiative. And we have a website here and a paper that was published in 2019 in BMC Biology. Then on slide 4, I give here the experimental design of this project. So here we are studying 4 different livestock species, which are cattle, goat, chicken, and pig. That are so the most extreme in terms of phylogenetic distance. So the distance is 300 million years. And for these 4 species, we have 4 different animals, 2 males and 2 females, on which many different tissues, like 40 tissues, were sampled and were kept. But in this project, we focused on 3 tissues, which are liver and CD4 plus and CD8 plus T cells. And for these 3 tissues, we are performing RNA-seq data for gene expression and transcript reconstruction, ATAC-seq for chromatin accessibility, and HiC for genome 3D topology. For the RNA-seq data, we are focusing on long and polyadenylated RNAs, where the sequencing was done in the directional way, and with a quite high depth of sequencing of more than 100 million paired unreads of 150 base pair per sample. Here I gave the genome assemblies and reference gene annotation. We are trying to use, when we can, the ensemble 90 annotation for almost all species except goat, where we had to use the NCBI annotation, since ensemble did not provide any annotation. And I'm going to come back on the pipeline, but we have more than 95% of the reads that are mapped, and between 60 and 75% of the reads that are exonic. So on slide 6, this is the RNA-seq analysis pipeline that we used. So basically, for each sample, we map the reads with star, and then we model the transcript using cufflinks, and then we merge all the transcript of the different sample using cuff merge in a novel gene annotation. From this novel annotation, we also detect long non-coding RNAs using the fill-and-see tool, and we also quantify the expression of both transcript and genes of both the reference gene annotation and the novel gene annotation using a STAR and RSEM pipeline, which is a pipeline that was used in many different consortia, such as ENCODE or Blueprint, etc., which is a good pipeline, although it requires quite some resources. And then we have all this quantification, long non-coding RNA, and also the novel models. And on all this, we also perform differential gene expression using EdgeR. And I've recorded this pipeline in the SNECMake language, and I give the link below. Thanks. So on slide 7, you have the number of reference and FR-AgENCODE transcript. I'm going to come back on what we call FR-AgENCODE transcript. So on the left, you have the number of reference transcript that we have initially in the annotation, and then the number of expressed ones. Immediately, you can see that if you look only at the annotation, you can see that you have not so many reference transcripts compared to human and mouse. In particular, at the beginning of this project, in this species, we had almost only protein coding genes, and then about 1 transcript per gene. So this is very low. Then you can see that even with 3 tissues, we detect between about 60% of the transcript. And then on the right, so what we call a FR-AgENCODE transcript is a cufflinks transcript that have TPM, so expression more than 0.1 in at least 2 samples to trust more these transcripts. And here you can see that we have many more FR-AgENCODE transcripts than in the reference, both coding and long non-coding. And I can come back on this later if you want, but if you, so we categorize this transcript into so coding and non-coding, and also into, we looked at their localization with respect to reference transcript, whether they were simply, they are exactly the same as an annotated transcript, or they were extending it, or etc. And actually, if we do all these summary statistics, we see that most of the FR-AgENCODE transcript represent actually novel alternative isoforms of reference genes, and they are coding. So if you go to slide 8, then we were interested in looking at the similarity between RNA-seq samples across our 4 species. So and this we want to do based on gene expression. So to do that, we have to get a common set of genes in these 4 species. So we have to focus here on the about 9,400 genes that are autologous between the 4 species. And we look at the expression in the different samples. So here on this slide, you have a heat map that is a squared heat map when on the rows and on the columns, you have the 39 RNA-seq samples that we have. In fact, you would maybe think that we should have, of course, 48, since we have 12 samples for animals and 3 tissues in 4 species, but some experiments failed. So that's why we had 39. And then the color, so the color of a cell is represents the similarity between 2 different samples, and actually the lighter, the higher the similarity. What you can also see on the right is a coloring, 3 types of coloring of the samples based on first species, tissue, and sex. And what we also did a hierarchical clustering of these samples based on the similarity computed as the Pearson correlation on the log10 expression. And what we can immediately see are 2 groups of samples. One type of sample are the liver samples, and then the rest, meaning that actually liver samples from 2 different species are actually closer to each other than liver sample from 1 species and CD4 samples from the same species. And then on the top, the CD4 and CD8 samples cluster actually by species first, and then by type of T cells. So that was this first picture. And then on slide 9, so we did identification of long non-coding RNA and try to quantify their conservation. So to do that, for the identification, we used the fill and seed tool that was developed in CNRS Friend by Thomas Derrien, and that is based on random forest to classify our transcript into coding and long non-coding. Actually, it has even 3 classes: coding, long non-coding, and a class of unknown potential, unknown coding potential. And here, if we do that, we see that a large majority of the long non-coding RNAs are actually intergenic, and it is consistent with what was found before in human. Then we can look for conservation. So as we know, the long non-coding RNAs are not very conserved in terms of sequence. So we have to do something different to identify their conservation. So what we did here, we are looking at sync-taining long non-coding RNAs based. So that means we look in different species. If we have 2 coding genes, I mean, if we have a long non-coding RNA between the same 2 coding genes in different species, then we call this sync-taining long non-coding RNA. And so we find 73 of them that are conserved across cattle, goat, and pig. And then if we introduce the chicken, which is further away, of course, we have less. 19 conserved between cattle, chicken, and pig, and 6 conserved across cattle, goat, chicken, and pig. And I think on slide 10, I've given an example of novel long non-coding RNAs that is conserved across many, many different species. You can see it on green here. It's the Chromos, we call it the Chromos gene because it is actually close to the CREM protein coding gene. And so you can see many, many species. And on the right, we have the expression of the 2 genes on the the two protein coding genes, and then in the middle, the expression of the long non-coding RNA. And usually, in most of the species, this long non-coding RNA is overexpressed in T cell compared to liver, but it's actually the other way around for the CREM gene. So it's quite interesting, this case. But we have other cases as well. Then on slide 11, so we use, as I said, the ATAC-seq to identify accessible chromatin regions. And this technique is actually much easier to use than the DNA-seq, since it starts from less material and is less time consuming in general, more easy to do. So basically, it takes the DNA and it will cut the DNA where it's open with an enzyme, which is a transposase. And at the same time as this enzyme cuts, it will introduce the oligo for sequencing. This is to identify promoters, enhancers, repressor, insulators, etc. And we did it using starting with about 50,000 cells with sequencing, so quite nice depths of between 50 and 100 million paired reads of 150 base pair per sample. But unfortunately, we had many mitochondrial reads, so we lose a lot, and we keep, after all the filters, between 10 and we only keep between 10 and 50% of the reads. I'm going to come back on how we did the pipeline, but most of the peaks that we find are intergenic, the large majority, and then intronic. Then on slide 12, so I give this pipeline. It's a very simple pipeline based on trim-galore for read adapter trimming, then Botai2 for mapping, then 2 steps of STAM tools to remove mitochondrial reads and PCR duplicates, and then the peak calling is done with MAX2. Actually, to gain power, we grouped together all the mapped reads from a given tissue, and we did the peak calling on this. So we obtained tissue peaks, and then to get a single set of peaks per species, we merged together all the tissue peaks based on a simple 1 base pair overlap, and we obtained between 75,000 and 150,000 peaks per species that cover between 2 and 4% of the genome. And these peaks, we were a bit worried that these peaks would be very large doing all these things, but actually they remain with a quite nice size of about 500 base pair. Then on slide 13, of course, we were interested to know if we had an accumulation of peaks around the TSS, transcription start site, and this is actually the case for the 4 species. We see the nice peak of ATAC-seq peaks around the TSS, but we have to say that still there is not the majority of the ATAC-seq peaks that are there. About between 10% and 18% are located there. The majority of the peaks are intergenic or intronic, and I have another slide where I can come back later. So then on slide 14, since we had this ATAC-seq and RNA-seq data, we were interested to see if there was a consistency, some expected behavior looking at ATAC-seq at the TSS and expression. We were actually thinking that maybe the more open the TSS was, maybe we would have a higher expression. So we're maybe expecting some kind of positive correlation. So we did that in many different ways, but here I'm going to present a way that I find interesting. So it's a per gene and a cross-sample correlation between the 2 assays. So per gene means that you take a gene and then you have the normalized values of openness and RNA-seq in the different samples. That means you need to focus only on the samples where you have both ATAC-seq that was performed and RNA-seq. And as I said, since some experiments failed, we do not necessarily have 12 of those. But there were 2 species where we had enough of this. It was in pig and in goat, where we had 11 samples for which we have both ATAC-seq and RNA-seq that were performed. So here I'm presenting the results on pig. And so for each gene, we have these values, and then we can do a correlation across these 11 points. And here I'm showing on the bottom some possible configurations. So you can have so a good correlation of 0.68, or even better in the middle, 0.92, or you can also have a negative correlation, minus 0.6. And what we do is we plot this distribution of correlation for all genes. So this is on slide 15. We have actually on the left the distribution of correlation for non-differentially expressed genes, and on the right the one for differentially expressed genes. And as you can clearly see, so for non-differentially expressed genes, you have a bell shape that is centered around 0. And for differentially expressed gene, you have a kind of two peaks, one at -1 and one at +1. And this was quite intriguing for us. We actually wanted to try. We wanted to see if this was already seen. We couldn't find really this kind of analysis done before, so we repeated it with roadmap epigenomic samples from human, and we actually had more curve that is more shifted to the right, and not really these two peaks. So for us, it is pointing to complex regulatory mechanism where you have both activators and repressors, but we want to investigate more this kind of thing, especially in other species, because we found this both in pig and in goats, and we try to eliminate all possible confounding factors, statistical factors. We worked with statisticians because I'm more bioinformatician, but this remains. So yeah, this is something to look at more deeply. Then in slide 16, for chromatin accessibility, we had to normalize the data and perform differential analysis as well. So we found that actually Nathalie Vialané helped us a lot with that. And she found that actually EDGER was not flexible enough with its TMM normalization to be used on ATAC-seq data. So we had to use the lowest normalization with CISO because it was more flexible, allowing one normalization factor per sample and per gene. And for the differential analysis, we actually used EdgeR. We had a model with individual and tissue, and when we used EdgeR, and here I'm presenting the results when we compare T cell versus liver. You can see that you have about the same number of more different over and under accessible regions, and between 5,000 and 12,000 differentially accessible peaks, depending on the species. Then if you go to slide 17, we were interested to characterize a bit more these differentially accessible regions. And so one thing we did was to look at the transcription factor binding site density in these regions, both for the differential and non-differential. So to do that, we simply took about 500 transcription factors from a JASPAR database, and then we just scanned the genome using FIMO, very simple ab initio approach, and we computed the number of TFBS in this region, divided by the length of the region, and we find that actually in red, the differential accessible regions have significantly more TFBS than the non-differential ones, so pointing to a regulatory functional role of this region. Then if we go to slide 18, so of course we have 4 species, so we can do many conservation analysis. So for this, we have to define conserved accessible regions, and so we did it quite simple, in a simple way, where so in each species we have these ATAC-seq peaks, accessible regions, and what we do in each species is that we simply project these peaks on the human genome, version GRCH38. And then, so if we look at where those projections are localized in the different species, and so we did a simple merge of this region, again based on one base pair overlap, and we call this human hits. And when a human hit comes from only one species peak, like the chicken, then we define this hit as having similarity level of 1. If it comes from 2 different species, a similarity level of 2, etc., until 4. And so the conserved accessible regions are the human hits that have similarity level, also called ontology level sometimes, more than 2. So then if you go to slide 19, again, we wanted to compare the differential and non-differential peaks with respect to conservation. So here we divided the ATAC-seq peaks in two different ways. First, according to their orthology or similarity level on the x-axis, 1, 2, 3, or 4. And also, for each similarity level, we can make two groups, the non-differential in green and differential in red. And on the y-axis, we are plotting the human heat FASCON score, which is a measure of conservation. So, of course, we have a correlation between, I mean, the higher the similarity level, the higher the FASCON score. It's normal. There are two ways of measuring conservation. But what is more interesting is that within each similarity level group, we can see usually that the differential ATAC-seq peaks are significantly more conserved than the differential, non-differential ones. So again, pointing to a functional role of this region. So on slide 20, so I put here a plot that I took from this review on the bottom to represent the different levels of compaction of the chromatin in the nucleus. So you have first the chromosome territories, and then inside these chromosome territories, you have the compartments, A for active, B for inactive. In active ones, you have genes that are more expressed, more accessible regions. And then within the compartment, you have these TADs or subTADs for topologically associating domains. And that are defined as the domains where you have more interaction within the domain than interaction between this domain and another one. And within this TAD, you have a finer scale conformation, which are these chromatin loops. And for example, in these chromatin loops, you can find the one that connect enhancers to promoters that are so interesting. And then on slide 21, so for this, looking at chromosome conformation capture, we use the in situ high-C technique that is indicated here, that comes from Rao et al. Cell 2014, that maybe you know, I don't know, but where you have the cell in the DNA from the nucleus, where you cross-link the DNA, then you cut with the restriction enzyme, you fill the ends and you mark them with biotin, you ligate, you purify and shear the DNA, and you pull down the segments that have this biotin, and then you sequence. And so, of course, when you map the reads, you do not expect this read to come necessarily from places that are close by in the genome. They could be far away, even on different chromosomes. And here we did that for the liver of 4 animals from 3 species, which are all but cattle, since actually it only succeeded in liver in our hands, and it didn't succeed in cattle. So unfortunately, but still we have these 3 species, and we managed to do it in the 4 animals with a very high depth of sequencing in each animal, about 280 million paired end reads per animal. So meaning about how did I compute 1.5 1.2 billion reads per species. On next slide, we have the pipeline that we used. So it's basically the high-C Pro pipeline. Yes, that was developed at Curie Institute. And then to characterize the TADs, we used the Armatus software, and we used an in-house tool for AB compartment. We'll come back on this. The normalization was done with the ICE. It's a matrix balancing kind of a normalization. And I can come back on all this IC analysis later on if you want. Then on slide 23, we do simple summary statistics on the read pairs that we have after different filters. So like the ATAC-seq, we lose quite a lot here, and the step where we represent the initial reads, then reported by the high-C Pro pipeline, then the valid ones, the one in good configuration, valid ones are good orientation, and then the valid after removing duplication, and then the trans, meaning on 2 different chromosomes. So we can see the step we are losing more reads is a valid step, which actually eliminates reads in inconsistent orientation, and we see a minority of trans interaction, and this was expected. So then on slide 24, the high-C contact matrix that we get are like this. So this we have a heat map where the more red the cell, the more read counts, and you can see clearly that the diagonal is highlighted here. But sometimes you have these squares that goes off the diagonal that represent these stats, and we expect these insulators at CTCF binding site in the regions where we have a boundary between TADs. So on slide 25, we wanted to have a measure of insulation strength of the TAD boundaries. So for this, we define, well, I mean, we were not the first one to define it, but we use this local interaction score that we computed based on a sliding window. So we move this sliding window across the genome of, I think we use 500 kb here. And we compute the number of interaction that cross the middle of the bin, and then the total number of interaction in this bin or sliding window, and we divide them. And we expect that at the TAD boundaries, we expect that this score would be quite low, because we don't expect many interaction to cross the TAD boundary if it's really a good TAD. So then on slide 26, we did exactly that. We had these TAD positions genome-wide, and we can compute both the interaction score in green and the CTCF density in red. And we clearly see that we have a peak of CTCF density at the TAD boundary, as we expect, and that we have this depletion of, this decrease of interaction score at the TAD boundary. So this was quite satisfactory, and since we were I think one of the first one to do this high-C, to perform this high-C data in some of the species, for example, goats, we were quite satisfied. Then on slide 27, we also did the AB compartment calling. Here, we look at the TADs and AB compartment and not the enhancer-promoter relationship because the sequencing depth is not high enough. So AB compartment is again a bit higher distance, lower resolution thing, but it was interesting also for quality control for us. So for this, we use for a given species the 4 bioreplicates we have. So we just simply add their counts for each bin pair. On the left, you have the raw matrix for species, and then we normalize this matrix with the ICE again software, and also we normalize based on distance, and we did that per chromosome. We obtain the matrix in the middle, and then so we compute A and B compartment a bit like how it was done in ITC software that simply does the Pearson correlation on the bin pairs, and then a PCA on the bins of the correlation matrix, and then the first PC, the sign of this first PC will indicate the boundary between, when it changes a boundary, it will indicate a boundary between A and B compartments on the right. And then on slide 28, we checked that we had actually higher liver gene expression in A versus B compartments. This is the case for all species. And on slide 29, the same from liver chromatin accessibility, which is significantly higher in A compared to B compartment. Then on slide 30, again, I didn't put a slide on this, but we could do this conservation analysis on the TAD boundaries exactly the same way as we did for the ATAC-seq peaks, projecting this boundary to the human genome and then computing this similarity level. And so for the ones that were in common between 2 different species, we could compare, we did the scatter plot of the interaction score in the 2 species, and we see a nice correlation, about 0.39 between human and pig, 0.42 between human and goats, and then 0.55 between our 2 livestock species here, pig and goat. And if you go to slide 31, we also this comparison between the orthology or similarity level of the TAD boundaries and their interaction score, and we see a clear decrease of the interaction score when the orthology level goes up. And this is very nice because the lower the interaction score, the higher the insulation strength. So this means that the town boundaries with more insulation, with higher insulation strengths, are actually also more conserved. So pointing to an important functional role again. So on slide 32, as a summary and conclusion, so I would say we have a highly consistent data set that I would say is quite exceptional in its design, in the sense that we have the same assays done on the exact same sample from some animals of 4 different species. Usually it's 2 species. You have 2 species and many more tissues, or you have 20 species, but you have only one kind of assays, like in the Villar et al. Data, for example. But having all this diversity of assays in all those species was quite rare. We have seen a complex regulatory mechanisms at TSS that you cannot reproduce with human data, but as human and mouse data will get bigger, we can maybe find, it's quite difficult to find these tissues where we have ATAC-seq and RNA-seq in different bioreplicates, so we can look. So this work represents an important resource for livestock, but also possibly for human and mouse that could be complementary to this data generated in this species, and everything, of course, is freely available and deposited in all possible banks where you can find them. We still need to do a better connection of this data to variation data, and it's expected to yield important knowledge for selection in those animals. And this is why the Gene Switch project was created, and currently it's a European project led by INRA that has started not long ago, that will run for 4 years, and that I'm involved in, and that will generate even more data, but on just chicken and pig. We will have RNA-seq ATAC-seq, but also promoter capture high-C, methylation, the whole genome sequencing, histone marks, and many different assays done on 7 different tissues in 3 developmental stages. So it will be quite interesting. And slide 33, I want to, of course, it is a work of many different people, sampling, assays, and data analysis. In particular, Sylvain Foissac, who co-led this analysis with me and did a lot of analysis, and Sandrine Lagarrigue and Thomas Derrien for long non-coding RNA aspects, Hervé Acloc for the assays, Nathalie Vialanet and Andrea Rao for statistical analysis. And I would like to thank you for your attention.